People with type 1 diabetes can no longer produce life-saving insulin because their immune system attacks and destroys their insulin-secreting islet cells. Replacing and protecting those islet cells from continued destruction has long been the holy grail of type 1 diabetes research. Now, researchers at MIT and Joslin are working on a method of sealing up transplanted islet cells in man-made porous protective packets, a technique called encapsulation.
So the idea of encapsulation is that you put a membrane around the islet, and the membrane's a little bit porous so that nutrients can get in, oxygen can get in, insulin can get out, glucose can get in, but the cells that are doing the killing cannot get in.
For decades, capsules have been made out of a seaweed gel called alginate. To create capsules, islet cells and the alginate materials mix, creating a liquid gel. That mixture is fed through a droplet generator, which forms alginate spheres a millimeter or smaller surrounding 1 or 2 islet cells. The spheres fall into a salt bath,
and that causes the liquid gel to solidify, to turn into a gel like Jell-O. And so it immediately captures that little droplet, and that's turned into a gel which is stable and strong.
These capsules produced good results in small animals, but the immune systems of larger animals identified the alginate as an invader and attacked the capsules, choking out the islets inside.
We're using what we get from the store. So you're putting an order in online and they're sending it to you, and every lab in the country and the world is essentially using the same thing. So we're limited by the materials that exist. So what MIT can do is take that store-bought alginate and make it into something better.
We needed something new, and we needed people who were experts in biomaterials, and that's what we've got at MIT.
What happened was the Juvenile Diabetes Foundation approached me because they were interested to see if we could come up with better polymers for encapsulating islet cells. That's really been one of the most serious impediments to developing a cell-based artificial pancreas.
We've really been taking sort of a biomaterials and engineering approach to this problem. We've really kind of stepped back and tried to figure out what are the best possible materials.
And the challenge when one makes a new plastic or one makes a new polymer is, unfortunately, it takes a long time. It takes, can take many months.
To speed things up, they use a process called high-throughput screening. A robot rapidly adds properties to the alginate that might help the material seem less like an intruder in the eyes of the immune system.
What high throughput enables you to do is to actually make thousands of materials, in a matter of days to weeks. So it tremendously accelerates the rate at which we're able to make progress.
Here it goes.
So what you see here are some of our actual automation resources that we have available to us. Specifically with our work with Joslin, we're interested in things that the body will tolerate to a very high degree without eliciting any type of immune response. The robot here can actually do hundreds or thousands of different pipettings or liquid dispense experiments, stuff that normally a person would do, only the robot never gets tired.
The MIT labs have already created thousands of new biomaterials, with features like controlled anti-inflammatory drug release and double-layered capsules.
Very imaginative approaches that I think is just what the field needs.
And so far, encapsulation experiments using some of these new materials show promising results.
Diabetes is one of the major medical challenges facing the world today, and I really do feel there's great value in bringing leading medical institutions like the Joslin Center together with engineering institutions like MIT to work on these problems.
It's not just about chemistry, it's not just about biology, it's not just about the engineering, it's about all those three things coming together to find a solution to the problem.
Well, our long-term hope is that if somebody had type 1 diabetes, that someday they could have an artificial pancreas.
Our complete expectation is that when successful, they will have normal blood sugars, no more shots, no more glucose testing, and normal blood sugar levels. Yeah, we're really excited about that prospect.